I'm Kayla, and this summer I participated in the John and Violet Kay Research Fellowship Program. When I was thinking of topics to research, I had no idea what to do. I kept asking myself, what could I research that I and other students like me could relate to and learn from? So I brainstormed ideas for a while until Dr. Chan informed Kelly and me about a new technology called iRepertoire, which would allow us to study the effect of something on the diversity of T cells. That is when I decided that something would be lack of sleep, because many students at Iolani School sometimes do not get enough sleep. I wondered if even just one night's lack of sleep would have an effect on the immune repertoire, which is why I focused my study on that topic. First, I'd like to start off by thanking Mr. K for supporting my research. The research question that I asked myself is, does my immune system become weaker when I do not get enough sleep? I know that whenever I do not get enough sleep, I feel very tired and groggy the next days, and I think many of you can relate to that feeling too. Many sources, including the National Sleep Foundation, recommend 9 hours of sleep for teenagers a night. However, many teenagers, especially at Iolani, often do not get the recommended amount of sleep because they stay up really late doing homework or other things. That is not good though, because there are many great health risks involved with sleep deprivation. For example, when you do not get a good amount of sleep, your risks of cancer, heart disease, obesity, high blood pressure, and even death increase. Although my research only focused on the immune system, sleep is vital for all parts of your body. There have already been many studies done on the effects of sleep deprivation on the immune system as a whole. One study by Benedict et al. Showed that the concentration of serum interleukin-7 increases significantly during sleep, which leads to the rejuvenation of the immune system. This means that if you sleep, then your body can restore itself. Another study by Bryant et al. Showed that the number of B and T cells decreased after 40 hours of complete sleep deprivation. This is because sleep causes the production of T cell-related cytokines, so no sleep leads to less cells. Finally, a study by Lange et al. Shows that adaptive immune responses are able to be more effective and powerful when you have more sleep. Overall, the message here is that sleep is very good for your immune system, which involves B and T cells. For my project, however, only T cells were looked at. So how is my project different than if there have already been so many studies done? The majority of the research that has already been conducted surrounds chronic sleep deprivation and extreme circumstances, meaning complete wakefulness for a few days. I could not do this without causing potential harm for myself, which is why I only stayed awake very late for one night. Also, the main thing that differentiates other studies from mine is that they all have only done studies on the number of T cells, and not the diversity of them, which is what iRepertoire can find. As stated before, iRepertoire is a company that uses multiplex PCR and next-generation sequencing to see the diversity of T cells in blood. The immune system is very complicated, as T cells do not just come out of nowhere. They have to undergo a lot of training to become a functioning cell, and then when the body finally decides that it is ready, it can go out into the bloodstream and fight. My hypothesis was based off of the other studies that I talked about before. Many of those studies only looked at the number of T cells and implied that more T cells meant better health. That is why I predicted that diversity would vary directly with the number of immune cells, because someone who has a greater diversity of cells has better health. To monitor our sleep for my project, my sister and I wore the Nokia Go Watch. It wasn't completely accurate, but it helped me to see my sleep patterns. Then came the painful part. On the nights determined by Kelly's menstrual cycle project, we collected blood from our fingers at the time points that I will talk about in the next slide. After pricking our fingers, we had to drop our blood onto the filter paper, let it dry, and then ship it off to the iRepertoire facility in Alabama. Also, to keep everything sanitary, we had to wash our hands and properly dispose of our hazardous waste. These are the blood sampling times that I chose. The timeline is not drawn to scale. On the night with 9 hours of sleep, I took 4 samples at 8 p.m., 2 a.m., 8 a.m., and 8 p.m. as my control. Then, for the night with lack of sleep, or 4.5 hours of sleep, I took samples at those time points, plus an additional one at 8 p.m. the following day, to see if there were any longer-term effects. After we mailed in our samples, we received the data back from iRepertoire and were able to analyze our data in many different ways, like in these colorful tree maps and 3D maps. For my project, I found it easier to just look at the D50 value, which is a quantitative value that is able to represent the diversity of T cells in a sample. These are the graphs of my D50 values. A higher D50 value signifies more immune cell diversity. On the top of my sleep graph is my hypothesis for how the immune repertoire would change with sleep. I hypothesized that the immune repertoire would restore itself at night and become lower in the morning due to previous studies. As you can see below, both of our data was similar to what I had predicted, as it went up at night and down in the morning for both Kelly and me. However, we do not know for sure if Kelly's immune repertoire went down in the morning, shown by the green circle, because there was not enough blood in her sample to rerun the data. Also, the colors are based off of Kelly's study, red for menstruation and blue for ovulation, because I had to see how my data was affected by her project. Then, for lack of sleep, I hypothesized that the immune system would not restore itself at night and then decrease even more in the morning. My data was somewhat similar to my prediction, but Kelly seemed more like my prediction for the sleep data. This is due to her first sample, shown by the green circle, so I could not draw any conclusions. The graphs on the right are mine, and then Kelly's data split apart, and they are color-coded based on my hand-drawn hypothesis graphs. Both my sleep and lack of data seemed somewhat similar to my hypothesis, but Kelly's lack of sleep data was a little different, so I was unable to fully draw any conclusions that could completely support my hypothesis. In conclusion, both of my data, sleep and no sleep, supported my hypothesis that the immune repertoire restores itself at night when you are asleep, and then it doesn't restore itself at night when you aren't asleep. However, both of Kelly's data had flaws and were a little different, so it could not support my hypothesis. Even if the data could not completely support my hypothesis that lack of sleep leads to a weakened immune repertoire, other studies have shown that lack of sleep has detrimental effects on the number of T cells and overall health. There are numerous other health issues, such as the risks of cancer and heart disease, that could increase with sleep deprivation, which is why sleep should definitely be prioritized. I would definitely do another project like this. Throughout the past summer, I learned so much more about the immune system and how to analyze data through technology. What was really exciting for me was I was able to take what I learned in biology in 9th grade, apply it to real life, and contribute to scientific research at the same time. I learned a lot during my experience doing research over the summer. I learned to not procrastinate, stay organized by putting my resources into Shobi and Mendeley, and so much more. I am so glad that I was able to choose a topic that I was genuinely interested in and wanted to learn more about. Thank you so much to Dr. John Han and Wenjing Pan for allowing me to use their iRepertoire technology, as I would not have had a project if it wasn't for them. Also, thank you so much, Dr. Chan, for being so supportive, helping me to create my research plan, and for keeping me on track the entire summer. I'm really grateful for this opportunity, and I really learned a lot. Okay, these are my references. Thank you for watching.